Welcome to the next lecture of this environmental biotechnology course. In this particular lecture, we will continue our discussion on the different methods which are applied in environmental biotechnology to understand the microbial community composition and their function. So, in today's class, we are going to address the following concept. We will discuss about the culture independent analysis method, but particularly today we will focus on amplicon sequencing based methods which are carried out through next generation sequencing approaches. So, the overall outline of the microbial community analysis within any environmental system, particularly if we are adopting a culture independent method, is basically the collection of the sample, and it could be as many representatives as possible. However, for simplicity, only one sample is actually shown here, but it could be as many as possible. And then extraction of the total community DNA or RNA. Now, when it is said that the extraction of total community DNA and RNA, it is to be understood that it intrinsically refers to the point that all the cells present within this sample are lysed and their DNA and if required the RNA are obtained, and a mixture of this nucleic acid is obtained. So, finally, that mixture of the nucleic acid, it may be DNA or it may be RNA as per our experimental decision, and then the PCR of the target gene. I will come to this point that why do we need to have a PCR reaction over here, and then once the PCR reaction is done, we get a product. And then one of the few methods that are available, that is basically the fingerprinting methods or the next generation sequencing based methods, are applied to identify and determine the relative abundance of the target gene, the different target genes. Now, as I mentioned, one intrinsic assumption in this case is that the sample will have many cells. So, when we take this sample, let us take an example that we are having the sample in a small tube and here we have the sample. So, we are adding our reagents. What reagent? These reagents are basically for cell lysis and DNA recovery. So, we add chemical reagents into the sample. So, this is the sample. So, this is the sample and this sample is ideally consist of many cells. So, there are many cells as I mentioned. So, all these cells are subjected to this chemical reagent like lysis reagent what you call, which actually degrades the cell wall and membrane and allows the cell cytoplasmic material and the nuclear material to be released into the solution. So, all these cells are broken and we get the DNA or the genetic material, the DNA or the RNA and even the proteins and also the metabolites released into the solution. Now, this solution is processed so that we get the desired molecules. Now, if we are desiring DNA, so we will have according the required treatment, so that only the DNA are the recovered. If we want to have RNA, then RNA will be recovered. If we want protein, or any specific metabolites with that will be produced. So, if we want carbohydrate or lipid, that can also be produced. So, these are all based on the reagents that we select. So, the kind of reagents. So, one part of the reagent is basically the cell lysis reagent, and another part of the reagent is the particular type of macromolecules that we are looking for. But the idea behind this the entire process is the extract the total. So, the importance of the total community DNA is very important. So, we must understand this very clearly, that here is no isolation of bacteria or no isolation of fungus, not that we are going to have separate cells with us and then the cells will be subjected to treatment, though that could be one of the options, but in general in situ lysis. So, this is basically called in situ lysis. So, we take out the sample, and in the sample itself we add the reagent, lyse the cells, and then we get the nucleic acids out of it. And then it is all about the chemical treatments, and those chemical treatments basically allow the segregation of the desired molecule like the DNA from rest of the molecules. And then once we have that desired molecule with us, so that is the DNA or the RNA. So, as I mentioned, if someone wants to work on protein or carbohydrate, that can be also done similarly, but that will be a different process or experiment altogether. So, if suppose we have the DNA or RNA, then we will subject this to a PCR reaction, because we want to study a particular gene. Now, once we have that particular gene PCR, so that the PCR product will be there, and then the PCR product will be subjected to the one or the more methods. Now, here are a couple of questions. Now, since this is having many cells, so the sample is having many cells. So, since samples are having many cells and these cells are of different species or phylotypes that we refer to them. So, we need to segregate or sort out these products. So, since we have actually taken the sample, we have the sample with us. From the sample we have extracted total DNA. Now, this total DNA is subjected to PCR. So, we have these amplicons, we will call. Let us say we have amplified the 16S ribosomal RNA gene. So, we have 16S rRNA gene from, since the sample is having many species. So, many species means many different rRNA genes. Now, interestingly, all these 16S rRNA genes are of same size. Why they are same size? Because we have actually used a set of primers which are always going to bound to a particular location, and then will allow amplification. So, we have same size, but they are different in terms of their base composition, that is their nucleotide base compositions are going to be different. So, basically they are different 16S rRNA genes. So, many different species. So, suppose if we have just assume that 100 different species are there, then there will be ideally 100 different 16S rRNA gene. Are supposed to be there. Now, how do we segregate these? So, these are actually some kind of expression that we try to do, that is the phylotyping. So, these 100 different types of 16S RNA genes are basically referring to 100 different types of phylotypes. Now, how do we segregate those 100 different phylotypes? So, for segregating those 100 different phylotypes, we know that there are a number of methods. We have already discussed in our earlier lectures that these can be segregated by clone library construction or a method like denaturing gradient gel electrophoresis, and then that is the physical separation of on the gel electrophoresis. And then there could be a very interesting method that we are very advanced method rather that we are going to discuss today is the next generation sequencing technology. So, all these methods are basically allowing us to decipher the different types of 16S rRNA genes which are in this case, because that is the gene that we are targeting in this case. But if someone wants to target some other gene, for example, the 18S ribosomal RNA gene in case of eukaryotes, or maybe some functional gene, any kind of functional genes which are very specific for any particular function, that can be also be taken. But we can take that assumption again that many different species will have many different version of that particular gene. That means the base sequences might be different. So, how do we segregate those different types or phylotypes? In this case, we are studying them. Now, conventionally, diversity studies, when we started with next generation sequencing method, before that most methods, that is the most molecular microbial diversity methods which are implemented in order to decipher the community structure in different environmental biotechnology processes, they relied heavily on the construction of the clone libraries. So, till the next generation sequencing methods were available and they were affordable, I will say, the clone library based methods were very popular, and till date many research groups like to use these clone library methods because they are easy to do. People already know how to go about that. You do not need the very sophisticated next generation sequencing machine, rather a simple Sanger sequencing machine which is more easily available in different parts of the country. So, they are still being used very popularly. But we will see today, we will discuss today that what are the drawbacks or what are the limitations of these clone library based methods, or even the DGG like methods that which we cannot possibly recover or we cannot overcome those limitations. So, ideally we should have some methods which are relying on next generation sequencing method, which are actually next generation term is used only to indicate that they are of advanced generation. Its generation means it is only the chemistry, the process, by how we are getting the sequence and the quality of the sequence, and it also includes many other parameters. So, I will discuss those issues, that why it is called next generation sequencing, but ideally it is a sequencing method. That means if you have a DNA stretch, you can get the composition of that DNA stretch, that is the ATGC based composition. So, what I am referring here, that molecular diversity research has used the clone library based method or the other gel electrophoresis based method like DGG for a long period of time, and still we are using those methods. But now we realize that there are certain things that we must be care of. So, one basic assumption in case of the clone library based method was this, that each clone in the library contained a unique sequence that was then used as a template for sequence determination. Now, most of the cases the culture independent techniques, as we know, bypass the need to isolate and culture microbes. So, we do not try to isolate any bacteria. Why we do not try to isolate any bacteria? We can always isolate a bacteria, and many times indeed that is being done for certain reasons. But if we are interested to know what are the species, what are the organisms present in an environment, if that is our question, that what are the species present in a system or a environmental process or environmental habitat, then isolation of bacteria may not be a very good choice, because isolation will lead to or give us idea about only handful of organisms or handful of or a limited number of members or the bacteria present in the environment. 99% of the bacteria or the species, they will not be isolated, and hence we will not be able to know anything about them. So, isolating and culturing them is not a very preferable way of doing it, particularly when we know that they have very serious disadvantages. So, in this case we are again coming back to this acid mine drainage, where a very highly acidic and heavy metal rich water is coming out, and I said it's a natural process, and it is basically catalyzed by very specific type of microbes along with the physicochemical factors, and it is contaminating the nearby areas unless and until it is treated properly. So, environmental biotechnology has a great scope to work on those type of environments, because in that case we can control the environmental deterioration, as at the same time we can recover the useful metals from those effluents or those water, that is called acid mine drainage water. So, what is actually done in this case, that basically we take the sample, that is a sample again, and then extract the total community DNA, and then this community DNA is subjected to some reactions which is called the PCR reaction. Now, as we possibly discussed earlier, that almost all the methods we see that these techniques, that culture independent techniques, they begin with the extraction of the DNA, because the DNA is the molecular biomarker. The macromolecule is a very stable molecule inside the cell, so and outside the cell also. So, we recover or try to recover the DNA from the sample, and or we sometimes try to recover the RNA also if we have some specific questions that I will come back, that, but basically it is preferred that most of the time DNA, but RNA can also be used. So, I write DNA or RNA. So, either of them can be or both of them can be extracted separately from the sample, and these actually will include this DNA or RNA, nucleic acid basically will include that the DNA and RNA from all the cells present in the sample. So, if we have a sample, so if it is having, let us say 1000 or 4000 different species, and each species having 10 or 20 or 50 numbers, so it is altogether maybe 5 lakh cells are there or so, or maybe little more or less cells are there. So, we expect that all cells are broken, lysed, and their DNA or the RNA is available to us. That is called the total extraction of the total DNA. So, we have protocols which are very well optimized protocols through which we can extract the DNA or the RNA, that is again total DNA or the community DNA we called it, from the sample. So, it is expected that these protocols will allow us to get the DNA and RNA from all cells. Sometimes we need to modify the protocols if our environmental samples are of any particular category or particular type. So, for that we need to consult the literature and see what type of modifications are to be incorporated. Now, after this extraction, what exactly we obtain? What we get actually is a mixture of all the DNA, and this is the genomic DNA mostly, and along with that the plasmid DNA will also be there. So, all the DNA present within the cells, and also the external or extracellular DNA, because if the cells are dead, then you have the extracellular DNA. So, all those DNA are basically extracted, and that extracted DNA is called, or the soup is called we call it genetic soup. Now, what exactly is genetic soup? The genetic soup is basically comprising of all the genes, because these are the short stretch of DNA which are functional, maybe producing the proteins, helping in producing proteins, or the ribosomal RNA or tRNA or different type of other RNAs. So, genes, genomes, transcript, that is the RNA, and transcriptome, that is the total pool of RNA from all the organisms whose cells were present in the sample. So, this is again a kind of a fundamental assumption that all the cells present in the sample are broken down, lysed, and all the genes or the genomes and all the RNA or the mRNA or different type of RNAs, that is the transcripts, and the transcriptome, that is the collection of all the transcripts, mRNA and RNA, they are in this genetic soup. So, genetic soup is basically a mixture of all the different type of genomes and genes and other things. If we have extracted the RNA, then possibly it is the mixture of the transcript and transcriptome. If it is extraction of the DNA, then we have the genes and the genomes extracted. Now, what do we do with this genetic soup? Now, a number of molecular techniques can be applied on these genetic soup in order to decipher, in order to answer the questions. What questions? The questions are to be framed by the researcher or the scientists who are working on this particular project. That, for example, one of the most fundamental questions is basically what are the organisms present there, number one, and number two is what are the phenotypic or what are the genotypic potential of these organisms, or what are the functional potential of these organisms. And the more questions like what are the actual functions carried out by these organisms, and also the interactions between these organisms and their environment. So there are four basic questions or fundamental questions that often we ask. So now these questions may be more fine-tuned based on the environmental process that we are working on. Now, what do we do with this soup? So we need to actually choose the methods by which through which we can actually answer to those questions. Now we have actually different type of techniques, as we already discussed, clonal library, DGG, or some advanced methods. So now the choice of the technique is basically depending upon the research question that is being addressed. So I mentioned 4 general questions which are fundamental questions, but these fundamental questions might be fine tuned, as I also mentioned, in order to make them more particular. And also it is also dependent on the temporal and the physical resources which are available. That means physical resources means what are the funds, how much money we have, because some approaches may be more expensive. So we need to actually take care of that fact also. And the temporal resources, that what are the resources available with the laboratory or the scientists at that point of time when they need the data, and also the monetary resources that they have with them. So based on that also. Now here are some important points to note. Frequently these approaches that we are going to adopt in order to decipher or get the answer of the fundamental questions of microbial ecology with respect to the genetic soup that we have just obtained. So these approaches actually target DNA. If we are extracted DNA, then they will target DNA, allowing the researcher to study the taxonomic and functional diversity of the sample. So most often these are the 2 fundamental questions, that what are the organisms present, that is the taxonomy, and what are the functional potential, that's the functional diversity. So we can actually target the taxonomic analysis, and also we can target the functional gene analysis. Now regardless of whether the DNA originated from dead, alive, metabolically active or dormant organisms, because earlier we have also learned that there may be different type of cells present. So since we have adopted some methods through which all cells are lysed, so all the DNA are available with us. So it's a mixture of all the DNA, and DNA won't tell us at this moment that whether the DNA is coming from a dead cell or a living cell, or was it a metabolically active cell, or was it a dormant organism who has actually given the DNA in our soup. So it is we have to take a decision and adopt appropriate method. So basically we need to target the DNA, and then the other questions we will answer slowly. So there is another alternative that I mentioned earlier, that RNA can also be used instead of DNA or parallel to the DNA. So when we use RNA, a clearer picture of the taxonomy and the functional diversity of the metabolically active components of the microbial communities can be obtained. So this will provide, of course, lot of advantage to us, because from scientific insight that actually what is happening. Why so? Because the RNA can be obtained only from metabolically active and living cells. So dead cells cannot give us RNA because there won't be any RNA present. So if we are targeting RNA, that means we are surely going to know that what are the metabolically active fraction of the community. So it may be a wastewater treatment system, or maybe a bioremediation field system. So in any environmental setup, if we want to know exactly which microbes are active, then possibly the best way to do that is addressing the RNA. However, these RNA based techniques have significant logistical challenges. What are the logistical challenges? That they are unstable. So RNA molecules are very, very unstable, and their extraction efficiency is also low. So if we are planning to have RNA based experiments or RNA based investigation about answering the questions and get to know what are the organisms and the processes going on there, then we must be careful about that. We must take appropriate care about their stability and also use a larger sample volume so that the extraction efficiency is taken care of. Now, once we address that issue, the next step in the sample processing involves the amplification of the extracted nucleic acid via the PCR reaction. The PCR has revolutionized the microbial or the molecular microbial ecology. So, as we have the basic understanding that the environmental sample is having multiple types of species and cells, and we can actually extract the community DNA or the RNA from that sample very easily. So, this is actually depicting the mixture of many different types of genomes, and that is part of the what you call the genetic soup basically. So, numerous type of DNA and the genome and the genes are present over here. Now, this is subjected to a PCR reaction. Now, this is PCR reaction, as you know, that is basically with the help of a set of primers. So, depending upon the set of primers, your amplification will go on. So, suppose we have used a primer which can actually amplify only 3 types of 16S RNA genes, then you will see only the red, green and the purple one will amplify. But if we use a set of universal PCR primer which can actually bind to any 16S ribosomal RNA gene. So, though there are primers which are called universal 16S RNA gene primers. So, they can bind to any, and you can have actually as many as. So, if you have let us say 4,000 or 5,000 species, so 4,000, 5,000 different type of 16S RNA gene. So, ideally 4,000 to 5,000 different 16S RNA gene products are going to be there. Now, this is basically very challenging task, because from the genetic soup to the target gene, because in genetic soup is basically mixture of all the genes and the genomes, and the PCR primer will bind to the specific location and will amplification. So, with as many as numbers are there in the genetic soup, we expect that so many numbers will be there in the PCR amplified product also. Now, the PCR process, since it is very specific, it enables the targeting, that is the target specific amplification primers of the genome or the transcriptome region, that is the gene of interest, to be isolated from the genetic soup. So, ideally what we can do, that the genetic soup might be having so many different genes together, all together. If we assume that there are 4,000 different bacteria or bacterial species, each having maybe 1,000 different genes, then 4,000 multiplied by 1,000, so it is basically 4 into 10 to the power 6. So, it is 4 million different genes are there, or some genes may be common. So, 4 million genes are there. So, out of 4 million genes, or maybe if we take into account the genes which are common, maybe there are, let us assume that okay, there are at least 10,000 genes which are unique genes are there. So, out of 10,000 genes, we want to amplify only one gene. So, that means it is possible. So, from a mixture of so many different genes and genomes, we will be able to pinpoint a particular gene of interest. For example, if I am working on, let us say, ammonia oxidation. In an environmental system, I want to remove ammonia. So, if I am trying to remove ammonia, I am targeting the organisms who are responsible for ammonia removal, that is the ammonia oxidizing organisms. So, in this case, there is a gene called AMOA, that is ammonia monoxygenase or ammonia oxidizing gene. So, if we target that gene, then out of so many genes, I said there could be 10,000 genes or there could be 50,000 genes together. So, out of so many genes, I can easily amplify the AMO gene. But again, the AMO gene will be having different sequence variations, because the AMO from different bacteria might have some sequence variation. So, those will be incorporated. So, that is a different question, but the point is that we will be able to have a targeted amplification. So, out of so many genes, our target we will be able to amplify. That is the beauty of this PCR reaction. Now, it has another advantage. The second advantage is that not only it will be very targeted, but also it will create millions of copies, that is called the amplicons. So, if I am targeting AMO gene from let us say a activated sludge or a wastewater treatment environmental DNA, then I will be able to amplify the AMO gene, that is one purpose, but it will be having millions of copies of that gene. that is the AMO amplicon. Like AMO amplicon means we will have millions of copies of or the reads of the AMO gene. Now that will help us to analyze the things further. Now the choice of the amplicon target, I have in this case taken for example the AMO, because my system might be working on ammonia oxidation. But if I am working on, let's say, sulfate reduction, then I will choose another gene which is called the DSR gene. But if I am working on methane production in a biogas plant, then I will select a gene which is called MCR gene. But suppose I am working on just on the diversity point, that what are the organisms present, then I will simply select the 16S ribosomal RNA gene. So the target gene that we select, the amplicon target, is largely dependent on the research question that is being addressed. Now, the challenge in evaluating the microbial diversity, either phylogenetic or functional, is now to separate the genetic soup of amplicon. Now, since we have, for example, amplified the AMO gene from the genetic soup, now AMO gene is not a single copy gene. I said a million copies of AMO gene, and if we had, for example, 4,000 different ammonia oxidizing species, so there could be many different types of AMO genes. So the amplicons could be of different types. So now we have to separate them. Now identifying the species, so for example in case of a microbial diversity question, so identifying the species present based on the differences across the amplicon of DNA sequence composition. So how do we do that? So suppose we have followed the same pattern, that from the environmental DNA we could amplify, and this amplification can be for the 16S ribosomal RNA gene in this case. So we can actually amplify using universal primer as many genes as possible. Now hundreds to thousands of different species will produce hundreds to thousands of different amplicons, or if we consider 16S ribosomal RNA gene, 16S RNA gene. Now DNA fingerprinting methods can be applied like DGG and all these things, and if we are planning to have a clone library, then possibly we can clone them individually. They will be inserted, and each of these clones will be subjected to further analysis by restriction analysis, that is the ARDRA, or maybe each clone can be sequenced directly. So once we do that, we will be able to identify each of these clones, and we will be able to identify most of these bacteria. So far their DNA is lysed and come into this soup, and they were actually amplified, because later we will discuss about that point. So clone library construction and sequencing remains a standard method for analysis of microbial community, phylogenetic diversity, and for assessing the functional potential, not only for 16S but also for many functional genes that we have discussed already in some other lecture, that a large number of genes are available which are very important or marker genes for different environmental relevant process. So based on or using those target genes, so clone library based methods were adopted, and it was very successful. So however, this process or these processes of clone library and then sequencing them are costly, time consuming, and lack the high throughput capabilities required for large sample volume. So they are costly because you have to first clone them, and then you have to sequence them, and also the time consuming, how many clones you are going to sequence. So that is a big question, because if we assume that there are many minimum 10 to the power 4 species or 10 to the power 5 species are there, that means 1 lakh different species or 10,000 different species are there, then at least few thousand clones are to be sequenced. So this is a very huge task, because all of these clones need to be processed separately. Their plasmid DNA need to be extracted, and then they will be sequenced separately. So they will be hugely costly and time consuming, because you need to handle so many clones without having any contamination between them. And for this process, the high throughput capabilities are very limited. Generally, they are not available for the common laboratories who are working on different aspects of environmental problems. So with handling the large number of sample volume, it becomes very inconvenient. Now here comes the next generation sequencing. Next generation sequencing is any of the several high throughput sequencing methods. So there are number of high throughput sequencing methods are there, whereby billions of nucleic acid fragments can be simultaneously and independently sequenced. So there are two things. So billions of nucleic acid fragments, that means, so if your sample is having, let's say 10,000 different species, and each species is represented by at least 10 cells. So that means total 1 lakh cells are there, because 10,000 different species, each species having 10 cells. So then the total number of cells are huge. Then that means when you extract them and you have a PCR amplification, possibly you will have billions of reads, which are called 16S RNA gene reads, if you are adopting that. Now, can that be sequenced simultaneously? Yes. In next generation sequencing method, we can sequence that amplicon, the pool that which comes from the genetic soup, can be sequenced directly. No need to physically clone them one by one and take individual clones, get their plasmids. All these things are not required. That is point number one. So it's a simultaneous sequencing of the billions of nucleic acid fragments. So it is actually 2 points. One is the billions of reads are simultaneously done. So its number is also very important. It's not 1 or 2 or 3 or 4. It's billions of reads are sequenced simultaneously and independently. That means you can have actually multiple samples together. That is also the case advantage. This is called multiplexing. So if you are using some kind of barcoding, so during your initial PCR reactions, then possibly you can discriminate the sequence products coming from sample 1 from that of sample 2. So those will be discussed maybe in little later in some other classes. Now that is something which is very interesting, and it contrasts the technique, the classical method such as Sanger sequencing method, which processes only one nucleotide sequence at per reaction. During the Sanger sequencing or the first generation sequencing, we are compelled to use a technology or a chemistry which handles only one nucleotide sequence at a time per reaction. But here per reaction it's the billions of different sequences, and these are some information about the different generations of sequencing, like the first generation, second generation and third generation. But some technologies are allowing short reads. That means the read of the sequence read could be like 500 to 1000 nucleotide, or in some cases that could be large fragments like several thousand, like kb fragments means kilobase. That means several thousand nucleotide at a stretch sequence can be obtained. Now these are some of the photographs of the sequencing machines. Now in terms of microbial community analysis, this next generation sequencing is going to be very important, and in the last 20 years or so there is a huge development in the sequencing technology which actually transformed the field of molecular ecology. While the use of clone libraries coupled with the first generation of sequencing technology, that is the Sanger, allowed only a small number of amplicons to be sequenced in a serial manner, next generation sequencing technologies are now allowed millions of amplicons to be sequenced in parallel. So this what I was mentioning earlier. In normal Sanger sequencing based method, you need to sequence one by one. Even when we have a multiple capillary Sanger sequencing machine, that is a sequencer, we can have a handful of samples can be run at a time, compared to the next generation sequencing where millions of samples, amplicons, can be run together. Now a major breakthrough provided by NGS approach is that the entire genetic soup that is obtained after the lysis of the cells from an environmental sample of the PCR amplification from the multiple samples can be pulled together and sequenced in parallel. So you can actually sequence the entire PCR amplified product out of the genetic soup, and also you can take multiple samples genetic soup. So if you have, let's say, 10 samples, so 10 samples genetic soups, they are subjected to PCR, and 10 now there are 10 amplicons. Those 10 amplicons can be run simultaneously in these next generation sequencing platforms, with the NGS platform directly separating out the different sequences type present while ensuring the sample they originate from. So they will give you read details, that is a 16S ribosomal RNA gene. If that is the target gene, you can always select some other gene. So the individual reads will be detected for each sample, and if you suppose you have taken 50 samples, for 50 samples the reads can be segregated. So it's a kind of an elegant approach. Now there are something more interesting rather than this only. So here is the picture that of the different environmental sample. We get the DNA and then allow the next generation sequencing method. So these are the different questions that often we get to answer actually. So, for example, who is there, that is the metagenetics, and what they can do, that is the metagenomics, and how many are there, that is the quantitative PCR, and what are they actually doing, the metatranscriptome. So, these three we will discuss later. We are right now discussing the first point, that is who is there, answering that fundamental question. So, we will come to this particular part of the metagenetics. So, some key points before we go to this, that the NGS methods do not require a cloning step. Individual DNA fragments are separated and amplified on the sequencing device itself. PCR products can be used directly for sequence. So, we use the genetic soup, do a PCR, and the PCR product, which is having actually millions or the more billions of different type of reads or the total reads, that is subjected to the sequencing machine. Number one. Number two is the volume of the sequence reads generated. So, as I said, the billions of reads are generated, and that is also connected to something which is called deep sequencing. Since hundreds of thousands of amplification reactions are conducted simultaneously on the NGS, the total number of reads are vastly exceeding what is possible through sequencing individual clones obtained in a clone library one by one basis. So, number one is that large number of entities, truly large. It is like close to a billion sequence reads that can be possible, and it is multiplexed. So, multiple samples together can be sequenced. Now, when we have several million reads coming out from a sample, so 1 million means 10 to the power 6 reads, which are equivalent to sequencing 10 to the power 6 clones. Otherwise, if we think of a clone library based method, and 10 to the power 6 means it is 10 lakhs. So, now if we have a 10 million reads, so it is a huge number of reads that we generate. Now, when we generate a huge number of reads, it is expected that the organisms or the sequences which are very rare in the environment, particular environment might have some bacteria or some archaea who are by number they are very less abundant, but they might be playing very important role. In a normal PCR and cloning procedure, we might have amplified them, but we might miss out them because their number wise they are so less that unless you sequence a large number of reads, you won't be able to catch them. So, the deep sequencing which is available through next generation sequencing, it is basically allowing us to identify the organisms who are part of the rare microbiome. Maybe I will talk about the rare microbiome in my next lecture. So, the metagenetics, that is an important tool for the biodiversity survey in environmental context. So, the issues that the metagenetics is going to address: first, the species identification and relative abundance. So, we will be able to identify each of the microbes present, because the reads that we are going to generate will be matched to a DNA database. There are beautiful database, very huge database are there. So, through the database match, we will be able to identify straight away that what is this read actually telling us, which is the organism for which is this particular read is. So, taxonomic affiliation will be there. So, that is our species identification and the relative abundance, because when we count how many reads belong to a particular taxa, we will be able to actually pull that information, that the relative abundance of the organisms. Second, to provide almost exhaustive enumeration of the organisms present, allowing characterization of the extensive microbial rare biosphere. Rare biosphere means the organisms which are less abundant. They may be more otherwise, but they are abundance wise they are very less. So, if you try to count them, they are not going to be counted because they are not generally present in so simple technique like a clone library or DGG. You won't be able to, unless you do a deep sequencing, you won't be able to catch them. Third is by targeting the specific microbial taxa, metagenetics approaches are also effective at screening for an identification of specific microorganisms, like a specific taxon specific 16S ribosomal RNA gene you can always use during your amplification process. So, you know that what organisms you are possibly looking at, and accordingly you could you can select the type of 16S primer which are important in the context of the specific environmental process, or you can actually target functional genes also, and this includes the contaminant removal. That means any particular organism who is known to be involved in some kind of contaminant removal. Okay, like the halococcoid is very important player in the hydrocarbon removal thing. So you can actually target such organism, or Thoira or Burkholderia or Pseudomonas type of organisms, which are often found to be very effective in degradation of different hydrocarbon compounds. Pathogen detection, like Vibrio and other pathogens, you can detect in multiple samples, and when you have large amount of data, suppose is in a sample, in a water sample, you have a particular pathogen, is a deadly pathogen, but the abundance of the pathogen is less, but still you can detect it. So that is called a sensitivity. Otherwise, in normal method, you would have said that no, this water is good for consumption, but actually the water is not good because it has the pathogen. Maybe the pathogen concentration, the load is low at this moment of time, but you never know, it may increase. So pathogen detection, particularly if the pathogen is at low level. Antimicrobial resistant genes. So if you want to target gene specific NGS studies, then you can actually amplify the antimicrobial resistance genes, and for different other purposes, human healthcare purposes, these are very important. And finally, for general environmental biotechnology processes, monitoring a particular process like the dissimilatory sulfate reduction, ammonia oxidation, methanogenesis, and many other processes, nitrate reduction, denitrification, nitrogen fixation, and so many processes which are very well known. Key enzymes are available. So you can monitor how the particular process is going on if you use such a primer in your amplification reaction. And it is also true that when applied to environmental DNA, that is called eDNA, so you extract the total DNA, so it can be targeted so that you can actually determine the presence of micro or macroorganisms that may be otherwise elude observation. So you can use actually 16S and 18S primer. Both primers can be used separately. You can actually then, for the same sample, if you use let's say if you use a bacteria specific, archaea specific and 18S primer, then simultaneously you will be able to check that what are the diversity and the organisms present in terms of the archaea, in terms of bacteria, and in terms of different eukaryotic organisms. So for this lecture, the following references will be useful. And in conclusion, I will say that the concept of the next generation sequencing based approach in community analysis is discussed. Particularly, we have tried to emphasize the need for the next generation sequencing. And although we talked about the different aspect of the common cultivation independent process, including the clone library based approach, but clone library was just one of the methods in order to address identification and the characterization of all the different phylotypes or the species who are present in the environment. And what we have learned that the next generation sequencing based approach is one of the best method in almost all sense. We will continue more discussion on this particular topic maybe in our subsequent lectures. Thank you.